A warm welcome to all the participants who have joined us for this 28th session of BISMiS Live. We have an excellent speaker with us who've been associated with BISMiS Live since the day it began about 3 years ago. And she's been working with BISMiS since the time of its inception. Giovanna has been affiliated with BISMiS for almost 10 years now. And for the past 3 years, she's been a regular attendee for the BISMiS live sessions, very active. Almost all the sessions, we've had one or the other question from Giovanna. But she's working at the Department of Biotechnology at University of Verona, Italy. She completed her PhD in agro-industrial biotechnology in 2004. And since 2014, she's been an associate professor of microbiology at University of Verona. Since 2018, she's also the deputy chairman of the subcommittee on the taxonomy of Bifidobacterium, Lactobacilli, and related organisms. And her research work, especially on the lactic acid bacteria, is an excellent piece of work. You can follow her website, lab website. You can read her research papers. And for her immense contribution in the field of lactic acid bacteria, in 2023, just about 2 weeks ago, she was awarded the International Science Award for Mid-Career Senior Researcher for outstanding excellence in lactic acid bacteria research with an industrial relevance. And from this award was given by the Lactic Acid Bacteria Industrial Platform. The website for this LABIP committee is labip.com. So you can visit there and see how this industrial platform is helping out in lactic acid research. So with this very brief introduction, I have the pleasure of inviting Giovanna to start her presentation and take the center stage.
Thanks very much, Kamlesh, for this very kind introduction. I will now share my screen to start the presentation. And as you mentioned today, I thank you all for this very important invitation for me. So I was very happy to accept, and I'm very happy to be able to share with you all the work that has been done on the reclassification, and but also on how we managed to involve stakeholders in this path that has been long. You will see it has been, Lactobacillus is a very old genus. It has more than, since the first description, it is more than 1 century and 20 years. So I will try to make it an interesting story for today. So first of all, a few words on Verona. We are in Europe. We are in the northeast of Italy. Verona is middle in between Milan and Venice. We are kind of famous because Verona is the location for Romeo and Juliet and other Shakespeare's pieces, but we are also a UNESCO heritage site. So it's a nice city to live in. Our university is quite young, and our department is even younger, but it is very active. We have 2 sites two buildings in 2 parts of the cities, and the department has been awarded this seal of excellence from the Italian government for the past 5 years and the next 5 years as well. And this is very important also for us today because we are going with the previous funding. We established a small culture collection, which is now funded also in the Next Generation EU framework. And these colleagues over here are really, with them, we are trying to build something new out of our experience in microbiology, and taxonomy is part of it as well. So as for today, I would like to share with you this path. And the presentation is divided into basically into 2 parts. One on the on the genus itself, so the path for reclassifications and how it is today. Because this is important to put names, especially, so the change of names. This name Lactobacillus is really part of the story of food microbiology and food biotechnology and probiotics. So it's a very important name. And when we wanted to change it somehow to, well, not to change it, of course, but to remove some lactobacilli from this genus and place it to other genera, we discovered many, many things. So we did it once in 40 years. The most frequent question that I got was, why do you want to do that? And so I will, in the second part of the presentation, I will try to make you understand what an interesting journey this was with many other people. So lactic acid bacteria, for those of you who are not familiar with them, they are gram-positive bacteria, and they are characterized, they are probably Lactobacillus is the most important genus for of this functional group of lactic acid bacteria that you will see referred to as LAB in the presentation. Is a functional group because this ability of producing lactic acid out of sugars is shared with other with different microorganisms belonging to different families and different orders sometimes. So in this word cloud, you see also the different sources of isolation of the type strains. So it's really related, they are really related to fermented foods, but also to environment. And I will give a little bit more details in a while. So yeah, as I mentioned, you find them and they are used as starter cultures or adjunct cultures in many fermented foods, also in probiotics. And their scientific impact from, if you simply, I did it yesterday, I did researches yesterday, you see that this name is highly cited in literature, in NCBI, in Web of Knowledge. So it's an important group of microbes. For me, it has been also a personal story because I spent the majority of my PhD working on lactobacilli and especially in producing 16S sequences to understand their phylogeny, because at that time, at the beginning of this century, this was not, there were not the 16S sequences for all the species. So at the LAB symposium, the 8th LAB symposium in 2005, I also brought this poster suggesting that maybe genus Lactobacillus was not a single genus, because Pediococcus, another very interesting and important genus for lactic acid bacteria, was always in between phylogenetically. So how can it be that two genera are one inside the other? So after this, there have been many things I will tell you about in a while, but we finally, you already know, I think, that we published these reclassifications in 2020. And so it was kind of a, closing the circle. And what is interesting here, first of all, I met many friends, I met many excellent scientists I had the chance to collaborate with. So it was really interesting also for this, but especially on the other side, on the scientific side, from the 16S insights, we were able to collect and connect all the information available based on genomes, based on metabolism, based on ecology, depending, of course, also on the expertise of the incredible team, the reclassification team that was in the end able to complete this achievement that is a citation classics today. Why Lactobacillus in an old genus? It has been described, as I mentioned, at the beginning of last century. But the big work on lactic acid bacteria was done by Orla Jensen, that published this book in 1919 on lactic acid bacteria. But here you see many different names And in bold, you see the subgenera of Lactobacillus that were characterized by different metabolic assets and so byproducts, also products and byproducts of their metabolism. I'm not going into detail of metabolism today, and I will stay more on the genetic basis for reclassification, because this is also the metabolic path is also very interesting and would take too much time for me today. So let's make now a big jump to the approved list of bacterial names, so 1980. And there were 36 validly described species names included in that list. So this is the beginning of the official nomenclature, as you know, but you already see this graph is taken from the reclassification paper, that's the increase of species description really increased a lot after 2000. So what I would like also to focus on in this talk with you today is, you see from 1980 here, we have different, every 2 years, we have a kind of numbers of species increasing. And you can see highlighted by the red stars, the years when species of Lactobacillus were reclassified in different genera. And the case I will talk to you about in a while is the case of genus Paralactobacillus that was described in 2000 and re-included in Lactobacillus in 2011. So it has always been a complex genus, and you have the idea here that some of these, the majority actually of these genera also belong to different families. So at the very beginning, it was a genus that really included many different organisms, and changing our approaches to taxonomy and systematics is helped us to reveal diversity in a different perspective and in a better way. Before talking about Paralactobacillus, I would like to focus your attention on this very important paper that was published in 1996 on polyphysis taxonomy, and it took also into account, sorry, let me change my pointer so it's for you to see, led by Peter Van Dam and Bruno Pot, that are also members of the reclassification team. I'm really happy about this. So here you see the phylogenetic grouping based on 6S and phenotype in metabolic. At that time, there were 3 metabolic groups. They didn't match. So you see the phylogenetic groups were metabolically heterogeneous, and also leuconostocs were a different group, including this big part of lactic acid bacteria biodiversity. In 1996, we had 62 species, and so we already see this heterogeneity, and that was already always the beginning of our papers. Lactobacillus is a very heterogeneous group, and so on, blah, blah. So let's see what happened for Paralactobacillus. In between, we have to remember that species description was always increasing. And the genus Paralactobacillus was described in 2000. And in this phylogenetic tree, you see this species and genus. The other phylogenetic groups, you again see also the Pediococcus together with Lactobacillus KZI group and the Bruchii here. That is actually Lactobacillus delbrueckii is the type species. So as now the genus Lactobacillus, the small one, I would say. And so the comparison was really made with representative species of the phylogenetic groups that were evident at that time, and was really important to observe some kind also of differential phenotypic characteristics with these groups. Reclassification of Paralactobacillus in the genus Lactobacillus was based actually on multi-locus sequence analysis, and again, phenotypic characteristics with the whole genus Lactobacillus, so not the phylogenetic groups, And the whole genus Lactobacillus, in between, there were the number of species were doubled. So of course the genus were even more heterogeneous. So for this reason, Paralactobacillus was in here. This is to give you an idea of the possible interpretation of this data having such a huge genus, Lactobacillus. So what's in there also? I mentioned that in 2000, actually, there is this increase in species description, but also the genomic era starts in 2003. The very first genome was this one, WCFS1, a strain of human origin of Lactobacillus plantarum that now has been placed in Lactiplantibacillus genus. Of course, at the beginning, the reference here, the reference list is really short here, because what I want to show you is this timeline where you have on your left, you have the number of species and papers that we published on the phylogeny, actually, to give an overview of the diversity of the genus. And on your right, you have papers trying to use the genome sequences that sometimes represented less species. Most of the time, not the type strains were not sequenced, but that were kind of trying to use the genomic insights to have information for the reclassification, to put some order in this biodiversity that was too big. The study that really changed this framework is this one. It was published in 2015, led by Polotul, University College Cork. And here, all the type strains for the species available at that time were sequenced, and the comparative genomics was made also with other with species of other genera, related genera of the family Leuconostocaceae, so closely related, but not the same family. So here you see from already from this genome study, and is reported in the paper, that the genus Lactobacillus appeared to be more diverse than a family, and also phylogenetically speaking, you see that Leuconostocs and Pediococcus were again inside the phylogenetic groups that were recognizable for genus Lactobacillus. Okay. So other researchers, Michael Genzel group and Jens Walter's groups, they focused on the same data, and they made a very interesting analysis of the genomes with respect to the lifestyle, and they hypothesized that different lifestyles of different phylogenetic groups. Again, here you see them. And also in between, you have seen previously 3 kinds of metabolism. And here we have 2 different metabolisms, because as I mentioned, in between also, there have been some changes in how to define the metabolism in lactic acid bacteria. So homofermentative basically use the glycolytic pathway, while heterofermentatives use the pentose-phosphate pathway. Okay, so there are 2 groups metabolically speaking, and inside those 2 groups, there are clearly recognizable some clades that could be an idea for also the reclassification. And this was the aim of this study that we published together with Polotul. And then also the Sarah Lebier's group in Antwerp, in Belgium, they joined this debate. And it was really interesting to share ideas and in the end, share efforts all together, and involving also colleagues of the subcommittee the subcommittee, the ICSP subcommittee, to have a consensus, to reach a consensus, and to suggest this reclassification that was really important in many aspects, as I will mention. A very interesting insight also came from the genome taxonomy database that was available already at that time. And really, you see here the screenshot that I took in 2019, so before the reclassification. You see the family Lactobacilli here, and you see the different phylogenetic groups inside there. And so it was really nice to be, and then started the comparison of who we had in the end to decide which species could go inside which genus, and then name those genera. So it was a long and an interesting path. But in the end, what we agreed on, the criteria, were the fact that these phylogenetic groups that represented monophyletic clades were to be the genera, the basis of the genera. Those genera was really interesting now because they shared ecology, mainly. They shared the same metabolisms and the main metabolic features. For most of them, it was possible to list exclusive genes or even gene families, so they were kind of different, and they were more homogeneous inside themselves than with the others. So really, here in this plot, you see in green, in yellow, and in blue the let's say the genomic homogeneity of well-defined genera. The genus Lactobacillus as it was before, so much more heterogeneous. The genus Lactobacillus and all the other genera as they are today. Again, this is in the reclassification papers, more details of that. So we were very happy with it, I have to tell you, because this is meaningful and could be also useful for diagnostic and identification of new families or new species in the genome, and I will explain a bit more afterwards. So we had to describe 23 novel genera. Then, of course, we revived the old Paralactobacillus name, and the genus Lactobacillus stayed the same and just smaller. So we had to find out 23 new names. Interesting story I'll tell you about later on. But also we had an interesting decision to make, either if the two families had to be kept separated or could be united. And the family Leuconostocaceae was described in Berg's manual based on 16S. We had also the evidence for the different metabolisms, but actually we thought that the reunion of the two families could be a better option to give also for the scientific community. And this is something that this framework, the description of novel genera and the union of the family, the two families, we think it's a robust framework that could be stable for many years. Of course, minor changes are still possible because there are some peripheral species that could, in the end, be the nuclei for novel genera, as it has happened in the past. But I hope and I think, of course, I'm not doing this again. So most of us think that this is a very robust framework that could be stable. And also again from genome taxonomy database, I want to comment a bit on the family, the decision of uniting the two families. I already mentioned this in 2019 with much more genome sequences. Also in genome taxonomy database, you see just one family level taxon. And I think it is very important also to make decision to have a consensus for different resources, because then other scientists use different resources, and it's really important, this coherency of information. among different important websites in taxonomy. So as for today, names and numbers, a bit of names and numbers. You have here all the genera in the family Lactobacilli. That means that we united the two families. So we have in total 33 validly published genus names that are here reported in order of decreasing number of species. Lactobacillus, you see, is still the biggest one. And we have, as of 1 month ago, we have 380 validly described species. Taking the list, so LPSN as a reference, but we could also see that there are some other names that are listed. Some of them are not validly published, but are cultured taxa, and some of them are uncultured, so they are candidatus. And you see that the numbers here change a bit. They increase, of course. In genome taxonomy database, again, there are 4 possible additional taxa, if I remember well, at the genus level. I'm not going into detail of this right now, but still is interesting to see that biodiversity that then we need to name, to refer to, is really big, and is increasing, continuously increasing. What I would like to mention with you today is a very short comment on this candidatus, Lactobacillus. So a new genus, and that was suggested in this very interesting paper based on chicken gut microbiome that reveal not only this new candidatus genus with 1 species, but 18 novel candidatus species of already described genera. So just the comment is simply this, that of course metagenomics, and in this case just a single group with 19 novel uncultured species could really contribute a lot. So it's really interesting and important to keep this in mind. So then this is the long short story made as short as I could. And now I want to share with you the impact and what we did also in the perspective of making this reasonable reclassification acceptable and accepted by the community, the LAB community, which is a very diverse and vibrant community. So it was really challenging, but also a very important and nice opportunity. And I have to tell you that I learned really a lot. So you already saw these figures, but what I want to focus on now is not only the number of publications you saw, but also the size of the market for probiotics, for instance. And this is an average of what I found in different websites of business intelligence estimates. So it is a very important research and development sector. And what I also have to mention too, and I don't know how familiar you are with this, but food and feed where lactic acid bacteria are mostly used is a very strictly regulated sector. And in Europe, we have this very important acronym that is QPS, that stands for Qualified Presumption of Safety, which is a concept described by the European Food Safety Authority. And it's really important because it's actually what it happens is that if you have a strain belonging to a QPS species, so it's a species included in the QPS list, you can kind of easily use it. If it's not, you cannot really use it, and you have to go through a very important safety procedure called safety assessment. Here in this pictures is taken from this very interesting review made for non-Saccharomyces yeast, so for other food-related bacteria. And what I want to show you is at the very beginning of the safety assessment, there is this taxonomic identification that has to be scientifically sound, of course, and the literature study, which means that you use names for searching the literature. And also EFSA, the European Food Safety Authority, when it revises this QPS list every 6 months, uses names and make literature analysis in title and abstracts. Okay, so this is very important to keep in mind. So taxonomists can suggest a change, but then there are all the other, of course, microbiologists. We are the same scientific community, of course, with different expertises. But also this nomenclature change affected a lot industry. Could also affect consumers, but actually is more in the, the technology transfer and in specialized media that I see stakeholders. And of course, the bottleneck for them could be the acceptance by regulatory agencies of this change. So really, stakeholders for taxonomy and taxonomic changes are many, and all of them are to be taken into account. But why, as scientists, we wanted to do this reclassification? Why this had to be done? Here is a very simple example of meta-barcoding data. So Amplicon sequences data sets of different fermented foods or human niches, analyzed with the old taxonomy, so the old Lactobacillus, or the new taxonomy. So you can easily see that in some cases, you can immediately see that Lactobacillus is the dominant or subdominant genus, the small one, okay? While in the other cases, you see that the former Lactobacillus was the name masking diversity. So it's much better now. We are much more able to reveal this diversity in the samples. And this ability to reveal diversity is also interesting because you can analyze and use microbiome data for understanding, for analyzing the distribution, the ecology of Lactobacilli in food and food environments. This is a very interesting paper published this year by Eugenio Parentel and this group in University of Basilicata in Italy. And this database is related especially to food and food environments. And ecology is part of the body of knowledge for inclusion in the QPS list. So it's really important also for applied purposes. For us, could also be very nice to use because we have the potential to investigate the distribution of genera of Lactobacilli, the small ones. You maybe remember it in the plot before, and you can see where they are distributed. So if you want to look for novel species, maybe you, at least from microbiomes, you can see where is better for you to go to be able to find novel species. So it's really interesting, and it's also always based on, genus names, somehow, the kind of analysis that you can make. But then also, another very important scientific perspective is the analysis of diversity for understanding probiotic mechanisms. So not only QPS, but also health claims. For supporting health claims for probiotics, what you have to prove is that there is a clearly defined mechanism. In 2014, Colin Hill and colleagues published this very important review on probiotic mechanisms, and the aim was to show that the different effects could be either assigned to some strains, sometimes to species more frequently, but then could be more widespread. To a taxonomist, this suggests that there are many different levels, many different possible taxonomic layers in between. So I'm really sure that reclassification is necessary to support further advancements. And so not only the prevent misidentification issues, and as I mentioned about microbiomes, but to be able to make meaningful comparisons, to try to reveal mechanisms and functions. And it is another very important review in the probiotic area published in 2018, so before reclassification, you see that different mechanisms could be associated to Lactobacilli genus Y, the biggest one, but Lactobacillus, some species. So what we plan also, and I'm sure people are already doing, is to see if this some species somehow relates, and we already know that it works, is related to genera. So QPS and probiotic mechanisms revisited where the reclassification could really provide novel frontiers for research. Okay, and this is for science and also application, but let's see the impact, the real immediate impact of this reclassification on the industry. We had to find a balance because it had to be an opportunity for all the stakeholders. It required a lot of discussions and a lot of sharing vision. So it was in 2020. What did we do before that? We presented, this is simply the list of what I or Elisa Salvetti did in this period. Other authors a little bit did something. But this was, we shared the idea, the diversity, the idea of a possible reclassification in many different conferences and roundtables. We were invited at the European Food and Feed Culture Association meeting in Milan. And in 2018, we also organized together with the Lactic Acid Bacteria Industrial Platform a workshop in Verona, when we brought together all the industries, but also that are part of this ABIP consortium, but also EFSA representatives, so regulatory agency, to make them aware of this change. We didn't share name of the genera. We didn't have them yet, but we wanted to, for them then to be prepared to this. And we published also the results of this expert meeting in 2019. Also, people present and others spread the verb, and we collaborated in a specific communication to the scientific and industrial community. And also, we realized, based on the suggestions that we got, we realized a converter of names. So old species name to new name, or the other way around. And this was developed by Sander Weitz. At the time, he was in Sarah Lebier's lab in University of Antwerp. And this website is still active and maintained both at the University of Antwerp and at the University of Alberta in Michael Genzer institution. And also, we realized the subcommittee, the website of the subcommittee, thanks to Paola Mattarelli, who is the secretary of the subcommittee. So where also people could have information on the member of the subcommittee. So this way of, make the change readily available, and also be as experts, be recognizable, so people can contact us and ask us advice on the use of novel names. This was something that we didn't talk about previously, but was really interesting. And also it required more effort, but in the end, it was really important for all of us to go through this all together. And this paid almost immediately, because thanks to this involvement and all this activity, what happened is that we published the change in April 2020 during the lockdown, and already in June 2020, EFSA, in the last update of the QPS list, included, maybe you can see it a little bit small here, but included this conversion table of QPS list, the previous class denomination and the new denomination. So EFSA and the European Food Safety Authority gave a reference also for other regulatory agencies. And this is really important for industries to have, a first kind of new benchmark for saying, it is in there. It has been accepted. There is no limitation in that. Another very important document we can refer to when we talk about microbial food cultures with safety demonstration is this inventory published by the International Dairy Federation. Is not only about dairy, of course, but is more in general for fermented foods. And they also included this new denomination in the last version in 2022. So in this perspective, the change now is completely in force, accepted, and with some important papers published that can give them industry reference to adapt to this change. Of course, I mentioned already that we have to find the balance, because on one side we had rules of nomenclatures to be respected, but on the other side, the nice thing of talking with so many people before reclassification is that we were able to understand their need. So of course, new names had to be stable, and this is also something that taxonomists want, of course. But there was a very interesting request to keep the initial L for genus name as much as possible, because the short form would stay the same. So then another kind of request, desiderata, let's say, was trying to find a way to connect the new name to the old Lactobacillus. So for instance, for this, we thought, and we did, that the majority of the names had Lactobacillus in the name, and we added a prefix with a starting L. So people, Levi, Lati, Limosi, there are many that had to be meaningful, of course, but there are many that have this idea behind. Then people complain because names are too long, but okay, you cannot find the balance with everything. But and of course, sometimes people not familiar with Latin can misinterpret. So Limosi Lactobacillus is not a Lactobacillus going on a limo, but is more referred to the ability to, of course, to produce polysaccharides. So it's slimy, means slimy, but it's all in there. It's simply that people has to get familiar with it. Or in other cases, we to keep the L, and to reconnect to specific phylogenetic groups, we suggested, and the nomenclature expert of IJSEM accepted this view, that Lactobacillus could be divided into parts. So lacti referring to milk, bacillus referring to the morphology and the old name, and casei or case, if you prefer the Latin pronunciation, and planti refer to the previous casei group and plantarum group. So I think that in the end, the strategy for naming was meaningful and took into account all the needs of all the stakeholders. So I'm coming to the conclusion, and I will show you, I need a few more minutes to show you what is happening now in the scientific community, because this is, of course, our community, basically, I think. And I want to go back to these two documents that I already mentioned. So the QPS list, this is the 2023, the last one updated, and this is the inventory. So the two documents I already mentioned. And here you see that the species listed in these two lists are much less than those validly published in the family Lactobacilli. So you see that this family that includes mainly beneficial or at least non-harmful but beneficial bacteria is still a big reservoir for food and feed innovation. There are many species that are not included in this list that are present in fermented foods, and we are studying some of them, for instance, in liquori lactobacillus or company lactobacillus that are not in those lists, but or only they are only in the IDF inventory, that could be used to improve the shelf life also of products because they are involved in fermentation. So that could be useful, to increase the shelf life of products and help us to meet the standard development goals, the zero waste, and improve the quality of food products. So you see, these numbers are important, 382 validly published. We already know that there are many others that could be considered possibly useful, and there are this list that give us an insight of possible. So it's really important for other species, and also for species present in one and not in the other, to reconnect with scientific literature, with patents, with genome sequences. So with all the data we are now familiar with. So following a discussion with a few colleagues, with Nicola Vitulo and Joele Lazzari of the computational biology group here in Verona, we tried to define a kind of R&D impact, research and development impact of Lactobacilli. Here you see all the different genera of the family, including the not validly published and the candidatus that I mentioned before. And then on the x-axis, you have the citations. And on the y-axis, you have the genome assemblies associated to that taxon ID. And the size of the circle is the number of patents connected to that taxon ID and genome sequences. So you really see that, of course, the species, the more they are on the right, the more impact they have, the more studied they are, okay? And so you see that there are few species really very well studied, and there are many that can be more studied in the future. So there is, again, a big potential over here. So I made an exercise. I selected 15 representative species of 7 genera deriving from the reclassification that are mentioned in the slide. And what I did was to try to see, so the reclassification was published in 2020, right? So the exercise was to reveal about the use of novel genus names in title and abstracts, because this is the approach EFSA uses. I didn't exactly use the procedure EFSA uses, but really quickly the same approach. And in years 2021 to today, so I gave these filters to the literature analysis. I used the Scopus, I used PubMed, and what I'm going to show you is the ratio between the use of the novel name compared to the total. When I put not new genus was to have, of course, to be able to verify that the sum of these two was the number that I got from the other, so the control. Okay, so guess and think about a number. Now I'll show you the numbers for those 15 species. Please, I see that I'm done with time, but I'll take very few minutes still. So here you have the numbers, the percentage. And here you have some species highlighted with the number of papers that resulted from the search. So you see that Plantarum is the model species. It has the highest number of publications in 2021, 2023. But still the use of the names is not, I expected no more, I have to tell you. The best performance is this on Lactobacillus curvatus, and it's still less than about a quarter. And there are some others that are really, sometimes depend a little bit on the numbers and the resource, but sometimes that scientific community working in that specific species didn't really started to use the new name. So concluding remarks, lactic acid bacteria are really key players in food biotechnology. And I hope that I convinced you, that I showed you that identity is really related and important to estimate applicability, which precedes application, of course. Reclassification has opened novel frontiers for research and development, and that has created some issues. Some had already were prevented and were mitigated for other stakeholders, but now we also have the scientific community a little bit that has to accept this change. But this renaming is really important to providing immediate ability to refer into homogeneous groups that are now the genera, and this is really important to boost research and innovation. So names are really important. We have to be aware that there is the big challenge of fragmenting information, especially when we have different resources and people not familiar with names. For instance, in food microbiology, an issue is also related to resources for bacteria and yeast nomenclature, for instance. And the big opportunity is that we can really make an effort to share the same language, not only information. So not only data, I would say, but information includes also same language, so names and nomenclature. And LAB community, please, if any of you is in the audience, please use and make others use new names when you are an author, when you are a reviewer. Please, journals, unfortunately, do not pay much attention, it seems, from the results of this, my very quick investigation. So let's try to make an effort all together to this. And just a couple, really few words for thanking all the incredible people that I've shared this path with. So the core people, it's very difficult to find a place where to meet all together. And members of the ICSP subcommittee were also part of the story. And Peter Van Dam was really important for the names as well. So it was really great in this. And Bruno Pot was really the one pushing the stakeholder inclusion in the debate. Paulo Tool, last but not least, of course, because he led this incredible path with us, and he was responsible for filling the gaps of genomes in 8 years ago already. And Paola Mattarelli here, who is the secretary of the subcommittee that was very active also for the subcommittee side too, and the website and so on. Being in the taxonomic audience, I would like to take the opportunity also to remember Professor Bruno Biavati, that sadly passed away during COVID pandemics, and he has been really important for bifidobacteria. Many species are named after him, so I think it's a very important thing to remember him today. And of course, I wish to thank what I call the Verona clade that was started by our ancestor and type strain, Professor Dell'Aglio, who is still, who is happily retired and was the chairman of the subcommittee up for almost 20 years. And he, together with Professor Torriani, they created this environment for taxonomy in Verona that is going ahead with me and with Elisa, who was making an excellent work also on safety aspects. And I wish to thank my computational biology colleague, Nicola Vittulo, and our PhD students, Ilaria and Joelle, that you saw mentioned on the slides where they collaborated with Professor Parente, University of Basilicata, and these groups. We have always very interesting discussion on taxonomy and how to implement this. And of course, the lactic acid bacteria industrial platform and all the stakeholders, because they were very active. Sometimes discussions were really, animated, but were always constructive. And I really learned a lot, and I'm really happy to have made this path. I thank you all for your attention. I'm sorry for the delay. I'll be happy to take all the questions that you wish to share with me. Thanks a lot.
Thank you, Giovanna. The talk was excellent, and I'm sure a lot of participants have questions. Some of the comments have come in, a lot of questions have come in. Aaron requested to make a short comment, so I'm going to enable Aaron to speak. Aaron, your mic is on, so you can speak before we take the questions.
First of all, this was a great talk. I enjoyed every word of it. I enjoyed the approach of naming your new genera also with the initial letter L to avoid the confusion. And I enjoyed the way that you coped with the, let's say, the QPS issues of the quality, so that the names are also acceptable in the industrial settings. I'm not an industrial microbiologist, and but I do realize that these names have also an impact on industrial settings. My comment was that I was very pleased to see that instead of splitting taxa in 2 or 3 or 4 more new taxa, finally someone decided to combine, to merge taxa, your Lactobacilli and your Leuconostocaceae, and make them in one family. It's a very rare case. It doesn't mean that Leuconostocaceae is not a valid name any longer. It still is validly published and it can be used, of course.
Absolutely. Yeah.
This in contrast to Lactobacteriaceae, which is an Orlayense name that I saw suddenly popping up in a manuscript I reviewed 2 days ago. So,
okay.
Seriously.
Nice to know.
Someone remembers these old names, but they shouldn't be used. They were not validly published. So that was about my comment, in addition to the fact that you and your colleagues cause a lot of work to the list editors of IGSCM who have to cope with listing all those names.
Yeah.
And maybe a last comment, and I was very pleased to see this, and I was not aware of it, and I will surely look into this very soon, that your subcommittee has its own webpage. I think it's the only subcommittee that really maintains an active webpage, and I'm very curious to look at it and to learn from it. And this is something that also other subcommittees may think of adopting.
Thanks, Aaron. Thanks very much for your comments. You are, you are one of my hero in this area. So having this comment from you I'm very glad. Thank you very much.
Aaron is a hero for everybody, I guess, in the field.
I think so.
Aaron did not make another comment, which he has written out into the question section. So he said that I somewhat fell in love with leuconostoc very early in my career, when as a student I grew big slimy colonies on sucrose agar inoculated with a sample from the sauerkraut fermentation. And these are interesting organisms that he loves to work with.
Absolutely, I agree.
Thank you, Aaron.
Thank you very much, Anna.
Yeah. I have a question coming in from Gerino Makori.
Yeah.
The question is, so thank you, Professor Felis, for your illuminating talk. My question pertains to the evolving landscape of assignment of new names, particularly in light of candidatus discoveries and the potential identification of new strains through metagenome assembled genomes. In your opinion, how should the scientific literature and community adapt to these advancements? That's the first part. And the second is, do you believe there's a need for new guidelines, or maybe there are too many already, or protocols to standardize how these discoveries are integrated into our existing taxonomic framework?
Yeah. Thanks very much for the question. Actually, I know Guerrino, so I know that is really, we have had some discussion on this as always already. And I think it's a very interesting and important question because, first of all, I don't know how we could adapt, but I'm sure we have to adapt. What I think, what this path has really made me more aware of is that we have to realize that on one side, taxonomy and, well, also the C code, so naming schemes that are interesting initiatives and are really important because they also demonstrate that the community is vibrant and the names are important, and this is the big thing. We are all the same scientific community, and we have to find a path in this. So if guidelines, or let's say the guidelines have to be issued, they have to be made by, not by few people, but they have to have a cooperative process. They have to be accepted. I was surprised. You saw the numbers in the scientific community about the use of new names. Okay.
Right.
We were all stuck in our houses for COVID pandemics. The paper was there. It was on the social media. It had been anticipated at conferences, but it seems that some people, they don't realize that their species, or they are not interested in changing names because they work on a single species and that's it. But your species is in a genus, and that name altogether make a difference. So also some taxonomic literacy for all the people is important. Taxonomists make decisions not to make life harder for others, but make decisions and suggestions to improve the working technical language of taxonomy. This is a citation from McGee comment 30 years ago.
Right.
So, but we have to be aware that there are many people talking. There are many papers coming out. Maybe what I think is that we should publish less and discuss more before publishing, because the taxonomic community has many ideas, and the freedom of taxonomic thoughts is something that we all believe in, I think.
Right.
But when it comes to people using taxonomy, they need, and especially I'm thinking about young bioinformaticians, that sometimes they are informaticians, not even biologists, and they have no training in taxonomy. So they analyze sequences in microbiomes, and they don't know where to look for, because they look at different databases and they find different things. So we have to maybe try to understand the needs of the community. And for instance, something that is really important for me as a food microbiologist, starting also a little bit to work on yeast taxonomy with experts, of course, because I'm not going to be, I think I'll never be an expert in yeast taxonomy, because it's really, of course, it's a different domain. They have a different genetics. It's a different organism. Everything is meaningful. And the tradition of having things treated separately, it's meaningful. But food microbiologists, they deal with bacterial names and yeast names, for instance, fungi names, fungal names. And when you sequence a microbiome, so Max, you encounter both. And not to say about phages, viruses, and taxonomic expertise is over there. So what I think is that we should be more aware of this as taxonomists. Maybe, again, publish important papers focusing on community needs. I'm a part of the food biotech community. There are others, and try to reunite perspectives,
right,
to be able to, not sharing only data, but really build knowledge out of data. I'm sorry, it was a long reply, but it's an important issue, according to me. Thanks very much for this question, Guerrino.
Thank you for that detailed answer. Actually, it does require a lot of discussion, because there have been many updates in the recent, past, especially related to, renaming of taxa, and then, inclusion of the phylum level into the ICMP, and then voting for using DNA sequences as type material, which was turned down by ICMP, and then seq code was established. So a lot of things have, happened in the past 3-4 years, where they significantly impact the community of not just taxonomy, but the entire microbiology.
Biotech.
Whether it is biotech, whether it is ecology, whether it is, anything else in clinical diagnostics or anything. And one of the biggest impacts of taxonomy, as a taxonomist, we feel that this is, the right thing to do, right? But then at the end of the day, if you look at the larger picture or the broader picture, especially in your last few slides, you have pointed it out very clearly that how well the taxonomic changes and the names have been adopted by the industrial sets. So analyzing the impact of your taxonomic reassignments is also very, very critical to see how taxonomy is impacting the fields. One of my questions that I wanted to pose was, although Lactobacilli are more important from the food perspective in terms of fermentation, there are very few that are known to cause infections in humans, right? So from the clinical perspective, Lactobacilli are maybe less important as compared to the food perspective. But whenever changes happen in the taxonomic field, they do have an impact in the clinical diagnostics. And my question to you on similar lines is, imagine that Lactobacilli had a higher prominence of causing infection as compared to being useful in fermentation or the food industry. How would you have assessed the impact of reclassification in a clinical setting as against a food setting?
It's an interesting question. Well, of course, other stakeholders would have been involved. I wouldn't say who, but I think that the same path to verify who could be impacted. I think that understanding who the stakeholders are and contacting them and explaining them why it is important to do this, that would be the part. With respect to the scientific approach, I would do exactly the same thing. And one of the things that I actually mentioned at the beginning is that I was going to focus more on the genetic and phylogenetic genomic part,
Right.
is exactly because this is more related to microbiomes and sort of genetic, to be able to genetically diagnose the species and the genus.
Okay? Right.
In the clinical perspective, I have other two comments on this, because when I started my PhD, there were some reports on the fact that a strain undistinguishable from a probiotic strain was found in as an opportunistic in an opportunistic infection, and so that questioned the safety of that probiotic. Okay, right. So diagnosis here is more at the strain level.
Right.
So, for instance, also the definition of a strain, what a strain is. There is a very interesting, very important review on interpreting strains in microbiome, published a few years ago by a Pierborg group. And we know that strain strain concept is complex, right? Right? So I think that phenotype there, if possibly of clinical importance, phenotype there would be, and the genotype behind virulence phenotype could have been more important, could be more impactful on this, because diagnosis in clinical setting is not only who is there, but how can I treat it, of course. So it could have had slight modifications. I think that the general perspective and the scientific approach for doing the reclassification would have been much more or less the same, but stakeholders, then specific inputs from stakeholders and taking reasonable time to understand and address this would be key.
Right. Right. I think this one is a comment from Christel Venter, an excellent example of how a community effort resulted in a much wider acceptance of taxonomic changes, and we need more of these. Thanks. Yeah. I think this one is it's kind of off the topic, but Naveed Abbas has a question. Ma'am, it was really informative lecture. Lactobacilli is an important group in relation to industrial products and industrial level, but I'm a plant taxonomist. And do you know of any other groups who are working on plant taxonomy? So kindly suggest.
I don't know, actually. Plant taxonomy is not my, no. Right. Outside of bacteria, I don't know. I'm sorry about this.
No, that's what I thought. It's kind of not relevant here.
Yeah, but you see, there is need for people working in taxonomy.
Correct.
So this is really something. So I would also, my comment is that, we need people working in taxonomy. We need to train a new generation of taxonomists, and a new way also to share information, and because it's not about approaches and papers, it's about people working, doing, collaborating, and sharing. So this is something very important, I think.
Yeah. One question that came to my mind when when I saw the table where you had now 6,500 strains in the new combined group.
Yeah.
Yeah.
The species, you mean? Yeah.
Yeah. So how did you find it? Was making it bigger more complicated as a single group rather than working into smaller split groups, as Arun also pointed out, that this is the first time somebody has tried to combine families, as against, the usual norm is to split, into smaller genera.
The family level, you mean?
Yeah, or the family level.
Yeah. Interesting point. We discussed about this. about splitting or merging, because we were splitting on one side and merging on the other side.
Right.
And I have three quick points on this. First, at the family level, we had very, very clearly these two metabolic groups that were more meaningful than the 16S about the families, for instance.
Right.
But then we, as experienced food microbiologists, thought about telling people that some lactobacilli were then in the family Leuconostocaceae. So people would have complained that, well, what, the lactobacillus is becoming Leuconostocs? Impossible, right? So that is again, a user perspective about experience in the group. The second is the genome, we could do both, but genome-based taxonomy for us, and also in genome taxonomy database, gave us the idea that they would be better to combine rather than keep them separated. That would stay forever because it's based on metabolism. Again, genome better than, well, there is also genome behind, of course, the kind of metabolism, but you know what I mean, the basic phylogenomic approach also, and genome taxonomy database was really, really interesting and important. And then it was also a practical reason for us, because when you look at lactic acid bacteria, they are in different families. At least we had the chance to put them together, not to forget anything in future comparisons. Because now Lactobacilli includes Leuconostocaceae always. And we have all the view of all these genera, and we never miss them again in comparisons. So it was, we had to choose, and we thought about what seemed to be more reasonable for ourselves and for users. Because also something that I didn't mention, that ecologically, when you look at species doing food fermentations, you always find couples of homofermentatives and heterofermentatives, one by one.
Right.
They often work together. So having them in a classification that kept everything in was more meaningful to us.
Right, right. So then it would be wise to suggest that in this particular case, the taxonomy, the science was accurate, but it was guided by its usability in the field.
Yeah.
Right? Yeah.
Absolutely, yes.
Yeah. So that's the point I wanted to make to all the listeners out there, that taxonomy, while working in the framework of the rules and, the physiology of the organism or the biochemistry of the organism, characteristics of the organism, it is eventually also looking at the usability in the real scenario, in real life sciences. And that's a perfect example. Thank you, Giovanna, for the excellent session and excellent discussion that followed. And I'm sure our participants must have enjoyed. And I'll look forward to your presence in the next one again on the next Saturday.
Thanks very much. Thank you all. Enjoy the rest of the day.
Yeah.